So excited that you can join us. I'm Julie Raskin. I'm the Executive Director of Congenital Hyperinsulinism International. I'm also an HI mom. My son Ben is 24 years old, and we're just so delighted that you can join us. We're going to have an incredible day today. And for those of you that don't know Congenital Hyperinsulinism International, we're a US-based organization with the global mission to prevent brain damage and death from prolonged hypoglycemia and to raise awareness of hyperinsulinism. We also conduct and support research for better treatments and a cure one day, and we support HI families all along the way. So we're so delighted that this is truly the world of HI here with us today. And we have a wonderful day today. We have speakers, we have breakout rooms, we have HI stories, videos from HI families, and much, much more. Ask the Experts panels and talks from biotechs and an HI status report. So it's going to be just an incredible day. And one little change is for the breakout rooms at 12:50. You're going to come in through the auditorium and click on the topic of your choice. Irrespective of what you chose before, please choose the same topic if that's where you want to go. But it's open and you just click into the room that you want. And now I want to thank our incredible sponsors for making this conference possible. Thank you to our sweetest sponsor, Resolute, and to our platinum sponsors, Crinetics and Zealand Pharma, and our gold sponsor, Hanmi, and our silver sponsor, Beta Bionics, and our bronze sponsor, Ami Bio, and I have to recognize the Chan Zuckerberg Initiative, because they are our sponsor through the Rare as One project. So they are also supporting this incredible convening and the work we're doing with the whole community to prioritize research and bring in the patient experience over a 2-year period. While you're at the conference, please visit the incredible booths that we have in the exhibit hall. We have booths from our sister advocacy organizations from around the world, and from the academic institutions and the HI centers and the biotechs. There's so much great information there. I had a really good time walking through. And the conference will be open all week, so you can revisit, you can come in, you can come into the auditorium, and you can see all of the presentations again, because there's so much great information there. And you can also visit the exhibits all week as well. So now it's my great honor to introduce a treasure in the HI community and my dear, dear friend, Dr. Davelyn Hood. Davelyn is the president of the board of directors of CHI, and she's also the principal investigator of the HI Global Registry. So Davelyn, tell us what you liked about day one.
Good morning, Julie. Good morning, everyone from around the world. Thank you for joining us. You know, if I said everything, that wouldn't be a fair answer, right? Because it was really a wonderful day yesterday. I loved seeing people through Zoom and connecting, even though we're not in person. I loved all of the chats that I had going on throughout the day. We're so blessed to have amazing sponsors and our experts, and they're so gracious with their time and so knowledgeable. I love that. But as wonderful as all of that was, I have to say, I am still singing the new CHI song. I woke up this morning, curled my hair, and was singing and bouncing all day. So everything was wonderful. But if you ask me my favorite, our new CHI song, I'm going to be singing for a while.
Yes, so Davelyn, this is what I tweeted about the CHI song. I tweeted, it's so catchy, but that's okay because we're virtual.
Exactly.
Yeah,
exactly.
Yeah, really
wonderful.
Yeah.
Well, good. Well, we have a great talk this morning to kick off today, day two. So I am going to take this opportunity to introduce another person who needs no introduction in our community, and someone I'm grateful has joined me here in Texas and many years ago, and helped with my son, who is now 19, but that is Dr. Paul Thornton from Cook Children's.
Thank you very much, Davelyn. I started my timer, so we should be good. Thank you, everyone. Yesterday was a great day. I really enjoyed all of the sessions. And today I'm going to follow on a little more in the area of updating everyone on what's going on. And hello to everyone around the world, especially my home country, Ireland. So hi there, people. Okay, so today I'm going to talk a little bit about what's been going on in the hypoglycemia world and really hit upon some of the points of advocacy that I and all of my colleagues in the hyperinsulinism field have been doing on behalf of patients with hyperinsulinism. So I am not seeing my screen. Are you seeing my screen okay, everyone? Okay, are we good now? All right. First of all, can someone help me? I'm not getting control of my slides. It won't let. Oh, there we go. Okay. So first of all, I want to say that we missed you all coming to Texas this year for the family event live. And here's a picture of our hospital. We would have hoped that we could have taken you over and shown you what a great place we have to work in. So what I'd like to do is sort of give you an overview of the changes that have happened in the hypoglycemia world over the last basically 20 years. And back in 2000, Kornblath wrote a nice paper talking about operational thresholds. And this was a guide to physicians to help them understand when do they need to intervene with newborn babies who are having hypoglycemia. And over the course of the years, there's been guidelines from the Canadian Pediatric Society. There was a meeting in 2009 sponsored by the NIH to help us learn what sort of knowledge gaps there were in hypoglycemia and its diagnosis and management. And out of that came the American Academy of Pediatric Guidelines. Now in 2010, the Danish put in place an algorithm to try and help their physicians avoid hypoglycemia and NICU admissions. In 2015, the Pediatric Endocrine Society put out some recommendations that were followed 2 years later by the British Association of Perinatologists. And in that guideline was, in many of our opinions, a little bit of a step backwards in the care for children with hypoglycemia. However, in 2019, the Canadian Pediatric Society came out with an update of their 2004 guidelines. So what is the goal of guidelines? Well, here's 3 of the guidelines. The Canadian Pediatric Society 2004 guideline, where they said that the purpose of that statement is to provide a consensus guideline that has practical applications for Canadian newborns and their caregivers. And in 2011, the AAP were offering a pragmatic approach to a controversial issue, which at the time, and still is what are normal glucose levels in the first 24 hours of life, which they said evidence was lacking, but guidance was needed. And then lastly, the Pediatric Endocrine Society set up their guidelines to help physicians recognize patients who have persistent hypoglycemic disorders, guide their expeditious diagnosis and effective treatment, and prevent brain damage in at-risk babies. So do the guidelines work? Well, there was a nice audit of the Canadian guidelines in 2009, and what they showed is that 1 in 8 Canadian babies who were born that year were screened for hypoglycemia. 1 in 30 babies born that year were treated. They found that 5% of babies who should have been screened were missed and not screened. They determined that the number of blood sugar tests on average for all the babies was between 5 and 6 blood sugar tests. And what they found was that the average age for screening was 3 hours of age, not 2 hours of age as per the guideline. So they made some recommendations on how to tighten those things up and move forward from there. Now, there was an audit of the Danish guideline, I think it was published in 2017 by Henrik Christensen, who's probably online. And what they found was that their algorithm caused a reduction in hypoglycemia following implementation of the hypoglycemia reduction strategy. However, they weren't able to prove it was directly related to the strategy, but the point was there was a reduction. So these were two good papers that looked at the guidelines and said, what has changed and what do we need to do to continue to improve? Now, a nice paper that also came out of Canada, and you'll hear Canada coming up a lot in this talk, was a review of changes in practice following the PES recommendations. And so published in Pediatric Child Health in 2019, they compared what happened to babies in their hospitals from 2011 to 2015, which is the period of time from the AAP to the PES guideline. And then after the PES guideline, they looked at 1 year to see what happened. And what they found was very interesting. They found there was an increase in the number of consults to the pediatric endocrine service, and that increased from an average of 6 per year to 33 per year. So the endocrinologists were being called in more often. And what was really important was that every single patient they saw had a serious cause of their hypoglycemia. All of them had hyperinsulinism, and then 2 had metabolic diseases. So this is very interesting because the implication here is that prior to that time, these babies may have been missed. And so their conclusion was that since the introduction of the recommendations, more patients were identified with real disease requiring treatment. And the suggestion was that this guideline helped pick up more babies. Now, there have been lots of papers published since 2015. So we're going to look at a few papers. I'm calling them the good, the bad, and the ugly because I'm a cowboy down here. So, and the first paper was a quality improvement initiative designed to reduce neonatal intensive care transfers from the newborn nursery into the neonatal intensive care for babies who are at risk for hypoglycemia. And this was published in 2018. And what they did is they looked at infants who are at risk for hypoglycemia, and then they put into place a project which was that we're going to get the babies to do bare skin-to-skin contact from immediately after birth, feed early, and have a blood glucose test by 90 minutes of age. And so this is dealing with the normal at-risk babies in the delivery suite and in the immediate time when they're moved to the newborn nursery. And what they found was a striking drop in the rate of babies who needed to go to the newborn to the NICU. So 17% prior to the implementation of this quality improvement project dropped down to 3%. So this is excellent because they were able to screen the babies and then decrease the number who needed to be admitted to the NICU and thus who were separated from their moms. Now, they also reduced unnecessary transfers, and their definition of an unnecessary transfer was someone who was sent to the NICU but did not need IV dextrose when they got there. And they dropped that from 5% down to 0.3%. But very interesting to me is there was no change in the number of symptomatic hypoglycemia babies. Now, how I interpret this was that screening newborn babies is going to result in us finding babies who have low blood sugars that are normal for their age. And this study was able to identify those babies and prevent them from going to the NICU. But this study did not prevent babies who got symptoms. So these are babies with real disease. So this is very encouraging because what they've shown is you can prevent the unnecessary admissions to the NICU, but the babies who need to go are still going to need to go. Now, another study out of Canada looked at babies who were not at risk for hypoglycemia and did not have screening done for the reasons of being either an infant of a diabetic mother or a premature baby or small for gestational age. So these are healthy babies who were not at risk for hypoglycemia, but who developed hypoglycemia in the immediate newborn period. And they found there were 93 cases in 2 years which turned out to be 1 in 8,000 births, that 78% of those babies had perinatal stress. And so you may recall that one of the differences between the Pediatric Endocrine Society guidelines and the others is that we recommend screening all babies with perinatal stress for hypoglycemia, and the other guidelines do not recommend that. And so this is sort of a little bit of a justification for that. 98% of them have symptoms, and of course all the guidelines recommend screening for babies with symptoms. But here's the symptoms: 35% jitteriness, 28% poor feeding, 25% hypothermia, 15% low muscle tone, 15% sleepy, 12% seizures, and 12% apnea. These are very serious symptoms and typically indicate a serious underlying condition. And so overall, 20% of these babies had major clinical signs of seizure, apnea, and cyanosis. And when they looked at their blood sugars, not surprisingly, they were much lower, 0.8 millimolar or 14 milligrams per deciliter, compared to the rest of the hypoglycemia babies who had average mean levels of 1.6 millimolar or 29. All of these are low, as you can tell, which is why they had symptoms. But interestingly, hypoglycemia was not detected until greater than 6 hours of life in the ones with serious compared to the rest. An MRI was performed on 14% of the babies, and 78% of the MRIs were abnormal, and 21% of the babies at the time of discharge had abnormal neurological symptoms. 52% had a workup looking for the cause of the hypoglycemia. So that's a little disappointing, that number, because we would like to see everyone having the workup. And in 37% of the babies, a cause was found, and the most common cause was hyperinsulinism. And they didn't discuss whether this was the genetic forms or the perinatal stress, but based on the incidence of the hypoglycemia, you'd have to believe it was primarily perinatal stress hyperinsulinism. So this was a very good study that showed the rate of problems in babies who weren't necessarily at risk, but by the PES guidelines, many of these babies were at risk because they had perinatal stress. Now, an article came out by Van Kempen in 2020 in the New England Journal of Medicine, and what they looked at was if you treat babies who have low blood sugar, either when their blood sugar is less than 36 or if it's less than 47, and you compare them to see how they did, which ones did better from a neurological perspective. And the reason they picked these numbers is because 36 or 35 or less is sort of the older value that's been recommended for intervention, and 47 milligrams per deciliter is a more modern value. And so they wanted to see how the changes in the interaction made a difference. So they looked at 689 babies. They found that their cognitive, intellect, and their motor skills were similar in both groups. And so they made the conclusion that in asymptomatic newborns, there's no difference in outcome if you treat babies when the glucose is less than 36 or less than 47. So of course, they did not say this, but the implication for people who just read the abstract of the paper, which is what unfortunately a lot of people do, is that babies with glucose in the first 48 hours of life do not need to be treated until their glucose is less than 36. So as you can see, there were a few of us who might have been disturbed about this. And it turns out that many endocrinologists and neonatologists wrote letters to the New England Journal pointing out some of the problems with this study. And the biggest problem with this study is they excluded 585 babies because they received IV glucose before the start of the study, or they had inborn errors of metabolism or HI. So of course, what they ended up looking at is babies who had that normal change in blood sugar that occurs in the first 24 to 48 hours of life, that most of us would agree does not cause harm. And they excluded babies who had real disease that most of us agree those sort of numbers can cause harm. And then they extrapolated that we shouldn't be treating babies unless they're less than 37. And so fortunately, your professionals, my colleagues, wrote to the New England Journal and pointed out the error of the ways in this paper. Now let's talk about the last study called the GLOW study, which looked at glucose profiles in healthy term infants in the first 5 days of life. So you would think this is a great study to do. Now we're going to find out what normal babies do. And so they looked at plasma glucose and CGM data for the first 5 days in a group of 67 healthy term breastfed babies who had no risk factors. So these babies were as good as you could get. They were purely breastfed, no supplemental feeds. These were the healthiest babies you can find. And what they showed was the glucose profile was very similar to what we'd found and others had found in previous studies, and that Dr. Stanley beautifully wrote about in his transitional neonatal hypoglycemia paper. And what they showed was that these babies normalized their glucose levels basically to adult levels by 4 days of age. And they confirmed the PES expert working group led by Dr. Stanley on normal glucose levels transitioning from intrauterine life to the world. So you would say to yourself, well, that's a great study. So why is Dr. Thornton upset about it? Well, here's why. Because in table 4, they put in a table that said, how many infants have episodes of glucose concentrations below the recommended threshold for treatment as recommended by the American Academy of Pediatrics, the British Association of Perinatal Medicine, the WHO, and the Pediatric Endocrine Society. And they came up with this table, and this data was presented at a meeting in front of 500 neonatologists. And when they showed the Pediatric Endocrine Society table and they said, you know, 50%, 70% of babies have blood sugars lower than normal babies, that the threshold for treatment in the Endocrine Society guidelines are lower than normal sugars in normal babies. There was a big old murmur of, yeah, well, I told you those guys were wrong. The problem is that the Pediatric Endocrine Society guidelines say nothing about normal babies. In fact, we recommend you don't test the blood sugar in completely healthy normal babies. Similarly, the American Academy of Pediatric guidelines say nothing about blood sugar levels in babies over 24 hours of life. So they were really comparing their normal babies to something that they had no right really to compare them to, and gave the implication that if this amount of 50% of babies have a blood sugar lower than the number the PES says, then maybe the PES is wrong. However, what they did not publish in the same paper is this poster that they presented at the Pediatric Endocrine Society, which was kindly given to me by Dr. Harris and Harding. And this shows several things: the blood sugar concentrations by age, and you can see in this line here that the dark line here is the average. This is the 10th percentile for blood sugars, meaning 10% of babies will have a lower blood sugar, and this is the third. And I think we'd all agree that anyone with blood sugars down here has a problem, and according to the PES guidelines, really needs to be investigated. And so what they didn't show in their paper, which is really not a good thing, is the beta hydroxybutyrate levels. Because where does beta hydroxybutyrate come from? It comes from the breakdown of fat, and fat, it gets broken down only when you're not having enough calories or when you're starved. So these normal beta hydroxybutyrate levels are less than 0.4 millimolar, which is down here. And you can see that the vast majority of these babies had low beta hydroxybutyrate because they were well-fed breast babies. But look at this, beta hydroxybutyrate levels going up to 2 millimolar, sorry. That's my 2-minute warning. That shows that many of these babies were actually, in fact, undernourished, and they're undernourished because the mom's breast milk didn't come in, which is not a problem in a healthy, normal baby because they build up their ketone levels and they're able to tolerate these blood sugars a little lower. But this is very different than the babies with pathological hypoglycemia, such as hyperinsulinism, who you all know are unable to make ketones. And so our babies have a whole different pattern. So this study really is being used to suggest that the target levels that the PES recommendations address are wrong, but the reality is that they're not wrong because A, we don't talk about normal babies, and B, these babies are able to make ketones, and that's a whole different picture from our babies. So to wrap up, there are different guidelines with different goals that have been published since 2000. The PES guideline was designed to help physicians diagnose babies with HI and other serious forms of hypoglycemia before discharge from the hospital to prevent long-term consequences of hypoglycemia. There have been multiple attempts to try and undermine the message, and so far, none of those attempts have been successful, primarily because your hyperinsulin experts have been reading these papers, writing to the editors, and publishing letters showing the problems in the papers. There's been some nice papers looking at the effectiveness of the PES guidelines in diagnosing the cause. There have been good papers looking at the effect of the guidelines on the frequency of blood glucose testing and length of stay in hospitals for all babies. And I would say that overall, the impact of the guidelines has been beneficial to advancing the science and understanding of hypoglycemia in the newborn. So I'd like to end up there and show you Peaks the Dragon. This is our hospital emblem. And this is a nice shot of the hospital at night. And Peaks the Dragon says, I wear a mask to keep others safe. So if everyone in Texas would follow that guideline, I think we'd be in better shape. So thank you very much, everyone. I'll hand back over to Julie.
Thank you so much, Paul, for your wonderful presentation and for the good guidelines. So it is now my pleasure to introduce a presentation from Christine Ferreira-Cook at Crinetics. So Jeff, I will let you take it away.
Good morning. I would first like to thank the organizers of CHI for hosting this virtual meeting and the opportunity to speak on behalf of Crinetics. For those of you who don't know me, my name is Chris Cook, and I am Medical Director at Crinetics, but a pediatric endocrinologist by training. I was fortunate to conduct my pediatric residency and endocrine fellowship at the Children's Hospital of Philadelphia, where I learned the complexities of treating congenital hyperinsulinism and the significant unmet need in this patient population. So who are we? This is a company photo which looks very different than previous years. Due to COVID, many of us have been working remotely, but we continue to be productive and have some exciting updates. Our vision at Crinetics is to be the leading endocrine company to develop new therapies for patients with rare endocrine diseases, with the goal of not only controlling their disease, but improving their quality of life. We have a robust pipeline with programs targeting acromegaly, Cushing disease, neuroendocrine tumors, congenital adrenal hyperplasia, and importantly, the SST5 program for the treatment of congenital hyperinsulinism. As we know, genetic defects in the pancreatic beta cell result in excess insulin secretion in patients with congenital hyperinsulinism. This leads to life-threatening hypoglycemia, and the hypoketotic nature of the hypoglycemia can have permanent neurological consequences. Our rationale for developing an SST5 agonist is that it would be potent at suppressing insulin secretion, but act independently of the KATP channel, and thus serve as a treatment for all forms of congenital hyperinsulinism, regardless of the genetic cause. The oral administration makes this ideal for neonates, infants, and children who suffer most. What I'm going to show you on the next few slides is preclinical data supporting that the SST5 agonist is effective at suppressing insulin secretion. The data here was conducted in collaboration with David DeLeon at CHOP. On the left is data from the SIR1 knockout mouse that has a defect in the potassium channel, similar to patients with KATP-HI. What you can see is that there's a robust insulin secretion in response to amino acids that is suppressed with the SST5 agonist. On the right is data from islets isolated from a patient with Beckwith-Wiedemann syndrome. Our SST5 agonist effectively suppresses glucose-stimulated insulin secretion from these islets. This slide shows data from our lab. The top two figures show that our SST5 agonist suppresses insulin secretion during an oral glucose tolerance test in rats, causing an increase in glucose. This effect is not only seen on day 1, but after 7 days of dosing, demonstrating the durability of our compound and the lack of tachyphylaxis. On the bottom left, rats were administered a sulfonylurea to close the potassium channel, cause insulin secretion and hypoglycemia, thus mimicking the hypoglycemia seen in patients with KATP-HI. Our SST5 agonist effectively reverses this hypoglycemia. Finally, on the bottom right is data from healthy human islets, where we again demonstrate the ability of our SST5 agonist to suppress glucose-stimulated insulin secretion. So what's next? We're finishing up all of our manufacturing and animal safety studies to allow dosing in people. We're going to move into humans. Our phase 1 study in healthy volunteers is planned for early 2021, and our phase 2 program in patients with congenital hyperinsulinism will start early 2022. But we want to collect more data. We want to better understand disease severity, duration, and family and patient concerns. We look forward to working with you on these exciting endeavors.
So great.
Can you unmute, Davelyn?
Yes, I can. Sorry about that, guys. So thank you to Crinetics for being a sponsor. Thank you for sharing some exciting updates as your compound is moving closer to clinical trials. And thanks for those that could join us. I now have the pleasure of introducing another sponsor to present information about their product, and that is Dr. Ed Damiano will be presenting from Beta Bionics. Ed, I muted you earlier, so do I need to unmute or can you do that?
Looks like I can do it myself. Thanks very much.
Awesome.
And shall I run video too, or no, I guess I'll keep that off. Okay, so I need to share a screen. Oh, you're muted.
You
I
can
can't hear you.
You can share your video if you'd like to, Ed.
Okay, it keeps saying I'm unable to start video, so that's okay. It's not allowing me to start.
Jeff, can you help out with that?
Now I can do it. I've got it. Somebody cleared it. Okay, so now I need to share screen, do I?
Yes.
Okay. So why is that not available to me? There we go. Oh, hold on just a minute. Just a minute. Be right with you. All right. All right. Are we live? Can everyone see us?
Yes.
Okay, very good. Thanks very much for having me. I really appreciate the time we can spend talking about the work we're doing at Beta Bionics with the Bionic Pancreas. My name is Ed Damiano. I'm the president and CEO of Beta Bionics Incorporated, a Massachusetts public benefit corporation. I'm also the professor of biomedical engineering at Boston University, and I'm the father of a 21-year-old young man with type 1 diabetes who was diagnosed at 11 months of age. So he's lived 20 of his 21 years with type 1 diabetes. So I just wanted to talk about what a bionic pancreas is and make it very clear that this is not a cure, of course, for type 1 diabetes, type 2 diabetes, congenital hyperinsulinism, cystic fibrosis-related diabetes, but these are all things we're interested in. And we do hope that this is a technology that could potentially be the next best thing to a cure until such time as a cure is found for CHI. So we have some compelling data in people with CHI that Diva De Leon has put together in collaboration with Stephen Russell to show us how this technology might work in CHI. The bionic pancreas, though, just so that if you're not familiar with it, it's a wearable medical device. It's about the size of a smartphone, a little bit smaller. It conveniently fits in your pocket, and it connects through a tubing, an infusion set with a continuous glucose monitor to your body, and it controls glucose levels autonomously in people with chronic conditions of glycemic dysregulation, type 1 diabetes being the first condition we've been focused on, but also other conditions such as CHI. It really consists of 3 device technologies and 1 or 2 drug technologies. It has a body-worn continuous glucose monitor. We use the G6 Dexcom for our technology platform. It has a wearable device that we've built. It's a dual infusion pump system. And it has a suite embedded on it of lifelong autonomous learning algorithms that dose insulin and or glucagon, either insulin only, glucagon only, or both insulin and glucagon every 5 minutes, using either analog insulin or analog glucagon, such as dasiglucagon developed by Zealand Pharma. Essentially, the technology adapts continuously and in real time to each person's ever-changing insulin and or glucagon needs. So this is a system that really is a piece of sort of connected digital health that is really sort of the quintessential of digital medicine, I would say, because it adapts to your ever individualized needs. And it's useful just to show you that we've been working at this problem for a very long time, first in animal studies, running on a laptop system way back in 2005, so about 15 years ago. And then we followed with adult studies and adolescent studies in type 1 diabetes at Massachusetts General Hospital. Did about 4.5 years of those studies in people with type 1 in the Clinical Research Center at MGH. And then in 2012, we built a wearable device that ran on an old iPhone, and at that time it was the iPhone 4S. It communicated with the Dexcom G4, and it controlled 2 insulin pumps, one to deliver insulin and the other was repurposed to deliver glucagon. And I should emphasize that we've tested various chronic conditions with this device, starting with type 1 in the home use setting, where people would wear the device for a week at a time or so, in adults and children with type 1 diabetes. We tested the system in the insulin-only configuration. We did other studies in the bihormonal configuration, and we did one study in the glucagon-only configuration in people with type 1. We've also done one study in collaboration with Diva De Leon at CHOP and Stephen Russell at MGH in congenital hyperinsulinism in the inpatient setting. Young people and children with CHI, post-pancreatectomy. And we also have done studies in cystic fibrosis-related diabetes, as well as in type 2 patients, adults with type 2, and also post-pancreatectomy surgery patients who have suffered from hypoglycemia. And finally, we built a dedicated medical device, which we call the iLet Bionic Pancreas, in homage to the islets of Langerhans. And we've done now 3 clinical trials in people with type 1 in the insulin-only configuration and also in the bihormonal configuration. But just turning back just for a moment, looking at some data from Diva De Leon's study that she did with the iPhone Bionic Pancreas at CHOP. You can see here each one of these lines represents data from one of the 10 subjects, with the red line being, solid line being the average for the whole cohort, and the red dashed line being the American Diabetes Association goal for therapy. And you can see that about 50% of people with CHI, these young people would have met ADA goal for therapy, but they would have spent about 1.5% of the time below 60 mg per deciliter under their own usual care. So this is, they spent a few days in the Clinical Research Center at CHOP on their own usual care, and then 3 days on the bihormonal bionic pancreas. Whereas about 70% on the bihormonal system would have met goal for therapy. You can see the mean glucose drops down. But I think really important for everyone out there is the reduction in hypoglycemia we saw with the system. The circles show you how much hypo each subject had. As you move to the right side of the graph and you look at on the right what the bihormonal system did versus on the left end of these lines what the insulin-only, what the usual care system did, you can see a reduction in the size of the circles, especially for people who had low glucose going in, and a reduction in hypoglycemia of almost a little bit more than 10-fold. So it dramatically reduced hypoglycemia in this population, while at the same time reducing mean glucose. And the importance of this device is that it's extremely simple to use. You just type in your body weight, and then it begins to autonomously adapt every 5 minutes to your ever-changing insulin needs. So you're not managing an insulin pump or a sensor-augmented insulin pump with a system like this. And that's really an important point. And then finally,
Thank you.
oh, I just wanted to point out some of the studies. Shall we stop now, or shall I
Yes,
just mention
but
this?
we're inviting you back for Ask the Experts.
Okay, very good. This is my last slide. Very good. Okay.
Thank you so much, Ed.
You bet.
Yeah. So now we are going to hear from Dr. Sunjae Bayak from Hanmi in Korea. And we're so delighted that Dr. Bayak came. It's the middle of the night there, but he joined us, and we are thrilled to learn about what you all are doing in Korea.
Okay, thank you, Julie. Thank you for everybody. My name is Seung Jae. I'm a head of clinical science and executive director at the Hanmi Pharma. And I know that everybody are not familiar with my company. So today I prepare some slide to introduce my company. And maybe you can see some our data about our last weekly glucagon on the first in human data, which was presented during this year American Diabetic Association. So please visit our booth and see the data and leave a comment and all the data to see the data. And you can see there is my screen, and this is the midnight around 12:40 at the morning. So yeah, this is forward-looking statement. And my company's name is Hanmi. We located in Seoul, South Korea, and we have some very strong R&D company, and we have a strong collaboration with the multinational company. And so, yeah, from the 2009, we have a multinational company collaboration. We license out our asset to Sanofi and Spectrum Small Biotech in the United States, and the MSD and Innovent is China Small Biotech. So for the rare disease, we have 4 assets for the rare disease. So one is CHI indication, that is weekly glucagon, and we receive orphan drug designation from the US and the EU. And we have another asset for the short bowel syndrome, is a monthly GLP-2, and we also receive orphan ODD from the US and EU. And we have some oncologic asset for the AML, which is very specific mutation in the AML, and also some enzyme replace therapy for the Fabry disease. And this is the last slide I prepared today. So this is our R&D pipeline. We are very focused on the diabetic and the endocrine or metabolic area. So we have a weekly insulin, which was licensed, co-developed with the Sanofi, and we have a weekly GLP-1, which is on the phase 3 with the Sanofi. So we license out our weekly insulin and weekly GLP-1 to the Sanofi, and it is ongoing development. And we have also for the GLP-1 and the glucagon combination dual agonist for the phase 2 trial. And we have another oncologic asset, and we have the other therapeutic areas of rare disease. So we have a weekly glucagon for the CHI indication, and monthly GLP-2 for the short bowel syndrome. And we have some rare disease in the eye ophthalmology area. So we have we also focus not only on the metabolic area and oncology, but we have put a high attention. My company put high attention on the rare disease area. So this is my end of presentation. So, and we just released the first in human data of our labs weekly GLP-1 at this year ADA, and we prepared that data in our booth. So please visit Hanmi's booth, and I will happily answer and stay at there. Thank you very much.
Thank you so much for that presentation. And like Julie said, for joining us at what is not a great time in your time zone. So now I am thrilled to share some information about the HI Global Registry. Again, I am Tai Pasquini, the Research and Policy Director here for CHI. And I wanted to start today off with a video that, although it's a few years old, we believe just really gives a great overview to the HI Global Registry, and also a little reminder of what it's like to be actually in person for one of these conferences. So I am going to share my screen now.
Ty, we don't hear the sound. I think it's
Oh.
because it's coming from your screen.
Oh, sorry. Let's try this again. Despite how many times you test, these things never go as planned, do they? How about now, Julie?
It's paused.
No, no.
Still no volume?
No, because it's, why don't you go on with your presentation?
I will. So I will remind everyone that we actually have this video in our booth, which we would love you to visit in the lobby. It is also available on higlobalregistry.org. So we encourage you all to take a look at another time, and sorry it didn't work, but we will just keep moving on. Can you see my screen?
Yes.
Great, beautiful. Okay, so I just wanted to go over some key terms and make sure we're all on the same page. So first and foremost, it's important to know that there's two key terms that you're going to need to understand when you're joining the registry. One is participant, which is going to be the patient or the person with congenital hyperinsulinism. And then the other is the respondent, who could be the participant, or it could be a parent, guardian, or a legally authorized representative. That individual must be 18 or older, but so you should understand the difference between participant is the patient, and then respondent is the person who will be answering the surveys. So the surveys all live on a secure platform that's hosted by the National Organization for Rare Disorders. And the study is approved through an IRB or an ethics committee. When individuals join the registry, they will be asked to share their experience through a series of surveys. And through these surveys, you're going to answer some questions. We'll tell you about some of the surveys in just a few slides. So then, once you've entered your data on this secure platform, the information can then be de-identified, and reports can be generated that we can then share with researchers. We will never share your name. We will never share information like your birthday or any other identifiable information. But when we take the information together, we can share it with researchers and clinicians to help the HI community understand more about the disease. And Davin will share a little about one such project happening right now later in the presentation. So why should you join? So one of the things we know is that a lot of people have questions, and we see all the time on the forum questions about if your experience is like someone else's in terms of what you're seeing with feeding or with other things that are going on. And in the registry, there are surveys that ask certain questions, and if there's a question about some of these things you have that you are wondering about, then you would be able to see a chart after you answer and submit the survey to show you who else, or not who else, but show you how other people have answered that same question. So you'll get a sense right away of how other people are experiencing their HI experience as well. All of this together also creates what's known as natural history, or the usual course and development of a disease. This can then be used in studies as a control group and helps us have a little bit more understanding over time, or more confidence that we understand the real experience or the experience of HI. And because HI is such a diverse disease, as we know with the different forms, the more people who answer and the more information we have, the better confidence we will have to help create better science and understand the challenges to increase quality of life and to lead to better treatments and cures. So how do you join? We want you to, you can join at higlobalregistry.org. There's a register button if it's your first time joining. And if you have already joined the registry previously, we encourage you to go back. The reason is that 2 of the surveys, pregnancy and birth, you only submit once, but many of the surveys are updatable, meaning that if something changes, you move to a new state, or you find out that you have a new diagnosis, or you change your medication, we want to know about that. And the surveys are designed for you to go back in and change or update the information so we can have the full picture. There are also a few surveys that collect data on a longitudinal basis. And what that means is that we want to, longitudinal is a research design that involves repeated observations of the same variable or question over time. So the more information we have, we can chart, and then we'll have a better understanding of that natural history over the course of time. So I also want you to know that I am here to help. I know it can seem a little overwhelming, but we promise once you get in there, it is easy. The questions should be an easy to understand language, and you can always reach out to me or other members of the team. And we really appreciate all of you who have already completed surveys and has led to some great insight, including in the 2020 annual report, which I will turn it over to Devlin to tell us about a little more.
Thanks, Ty. First, let me just tell everyone that for the last 4 or 5 years, when we've talked about the registry, you've seen me presenting the information, and I really have to just welcome Ty with open arms. She's new for us this year, but has really hit the ground and gotten up to speed. So a big thank you to Ty, and I look forward to continuing to work with her. One of the big projects we started with, which was probably more than I could imagine, just starting in the middle of a pandemic and everything, was getting this annual report done. I included this link here, if you can see at the top, where you can find the registry, find the annual report on the CHI website. Ty, we can go ahead and go to the next slide. Let me orient you to the slide real quick and start with the title. That should be obvious, but what you will see is a date. Although we have some updates that we know of internally, I'm basing everything from the registry annual report this year. So a lot of that was data that we pulled at the end of January or first part of February. So you'll see the date as to when this data is accurate or when it was pulled. So as of February, we had 303 participants added to the registry. And all but 40 of those participants that have been added went on to complete the consent process. We had about 3/4 have started taking surveys, and we've had, or meaning when we say started or begun taking, they've completed at least one survey. But only about 1/3 of people have completed a full set of survey questions. The positive note, however, is that we have 44 countries represented. And Ty, if we can go to the next bit. Also interesting, you see a very wide age range. Our youngest, as of the beginning of February, was 5 weeks old. The oldest was 58. The majority of our participants in the registry are 5 years or younger. But we do have a very nice cross-section of the HI community to kind of capture what life with HI is like across various stages of life. Ty presented the different surveys. Here's just a brief look at the number of individuals as of February who had completed each one of those surveys. So we can go to the next slide. Again, we highlighted we had people from 44 countries. What you see at this point here is where those are located, and we have individuals represented in HIGR from every inhabited continent. However, we do see a bulk coming from English-speaking countries, and I just saw a question about will the surveys be presented in different languages. I actually just spoke with NORD this past week. They assure us that they continue to work towards having the platform in multiple languages, I can also assure you that that is something that I ask every single time I'm on the phone or in person with NORD, is when it will be available. So it's a big priority for us, and at the moment there's work that continues to go towards getting it in multiple languages. Over half of the participants in the registry report having diffuse type of HI for focal disease. We feel is likely underrepresented at this point, and there's a number of reasons we won't go into at this point. And we do have about a third of participants reporting having an unknown, undiagnosed, or suspected, but not yet confirmed type of HI. Again, what we're trying to present is HI from the patient and family perspective. So we lump all of those together in a not specified component right now, or a piece of that pie, and there's really worth, it's really worth digging in there at some point and understanding what that means. And it's something obviously we hope to do in the near future. So we can go to the next one, thanks. One of the sections that we do feel is quite strong in the registry is the medications that have been used. Particularly, we have a large number of individuals who have taken the older drugs that have been around to treat HI, specifically diazoxide and octreotide. And Ty, if we can click one more time, thanks. So in the annual report this year, we noted both the side effects of diazoxide and octreotide, and they were actually very, very similar to what we reported in the early glimpse report last year. Basically, the top 5 side effects were the same year to year, and we just saw some slight shifts in what order those top 5 were in. One thing that, not quite yet, Ty, thanks. One thing that we do want to highlight here, and it's a big part of the education and advocacy work of CHI, and so many of the others that are on this the presentation or part of this our meeting this weekend, is that regardless of these 2 very well-known treatments for HI, continued hypoglycemia tends to be one of the more prominent side effects or lack of effects in this case. And so it's really, really important that what we have currently, that individuals understand, that the drug makers understand, again, that the FDA and the EMA understand, is that our current treatments are not effective for treating HI. And so we're so grateful that we have a very rich pipeline and number of sponsors represented here in the conference this weekend who are working on new treatments. So this is a very busy slide, but it highlights a couple of important points that I would like to make about the registry. One, and we touched on this yesterday, and so I was excited to get to present this today, but feeding issues are very, very common in HI, and in fact, over 2/3 of children with HI report having some type of feeding issue, and then we break it out as to what that type of feeding issue was in the green section. There's a lot of further sub-analysis that we have planned, but that does kind of point me to a couple of things. One is you can see, regardless of type of HI, we're seeing feeding disorders or feeding issues. We're also seeing it not just related to the type of treatment. So it's not those that have pancreatectomies and those that don't. So it is important, and there's a lot of sub-analysis we can do. But sub-analysis here is like this one. It takes responses to all of the relevant survey questions. So here, if you remember from the the first slide, we had 118 individuals complete the diet and feeding management survey. But this question actually, or this analysis takes 3 questions. It takes the feeding survey, it takes the surgery survey, and it takes the HI type question. So it's really, really important. And then what you see is that what we started with 118, we dropped down to 98. And so this is something that I just want to encourage those of you who have begun taking surveys, you've registered and consented, and you've started the process. Completing that is really important. And we can go to the next slide, Ty. So here's another example of that, where we have 116 individuals who completed the surgery survey. 43 individuals or participants report that surgery was considered for their management of HI. We lost a few here in that we only know more information about 39 of those 43. 8 chose medical management, 31 report undergoing surgery for the management of HI. We're going to focus on diffuse and focal here just because there's such a small number in the atypical and not listed type or HI type. So of focal, and Ty, you can click one more. There were 9 that are included in that bucket, and 19 were diffuse. And so essentially, again, there's so many ways to do sub-analysis here. This is just trying to give you just a very high-level sense of the type of patients that we're seeing in the registry. What you can see in the case of focal and diffuse is, essentially that the majority, the vast majority, but not all, of children are having their surgeries within the first couple of months of life. Great. Another really rich section of the registry is the quality of life section. We have over 100 parents represented here. And in this case, the way the registry works is only 1 parent per participant is able to answer the parent quality of life survey. This year, the biggest growth that we saw was in the primary school age, ages 5 to 8. And you can see from the graph, though, that's on the screen, that we actually have parents of all age groups represented. So if we pick just one question, and Ty, you can do the next one, thanks. All of the questions had a very similar trend in quality of life, but we'll look at this one where parents are reporting how they perceive the HI management by age group, and we lumped them in slightly different age groups here. We have the under 5, we have 5 to 12, and then we have teenagers and above. In that situation, what you see here is that if we look at options like disruptive, which is the gray bar there, parents of older, fewer parents of older participants report that adjective than of younger parents. And then likewise, if we were to look at simple, we have fewer younger parents reporting simple than of older parents. So we don't dig a lot just yet into why these trends are there. And we hope that this does give younger parents of young HI kids some hope. But I don't want to leave the slide without recognizing the fact that across all age groups, every one of the options are present. So you still see for 13 and above disruptive, you still see simple happening in the under 5 age group. So it is important as we look at further into the whys and hows of this, but in general what we're seeing is that, and it's something kind of anecdotally we've known, that it does seem that the management of HI does improve over time. All right, so that's data. You know where to find the report if you want to read more. And here's what we have as a call to action to parents and those with HI. First and foremost, we want you to register. And Ty had shown you where you can do that. And once you register, you're going to be asked if you would like to be contacted for certain things. Our desire is that you say yes to everything that you can be contacted for. We do break this out in the report as to what that means, but you have opportunities to be contacted by us if there are, by us at meaning the HIGR team, when we have questions or new opportunities within the registry itself. You have the opportunity to be contacted if there's a clinical trial that you might qualify for. And you have the opportunity to let us know if you want to be contacted if there are other networking opportunities, such as your region is getting together or other HI types similar to what your participant has, are choosing to get together to do something as far as connecting in some way. So we definitely hope that you say yes to those opportunities. We want you to consent to participate. It's a long document that takes a little time to work through, and if you agree, you consent to participate. If you have any questions, please reach out to Ty, and she can walk you through that. As I had mentioned, we want you to complete all the surveys, update them, and complete the longitudinal when you have a chance. All right, so what are we doing next? We are hoping to begin to fill the gap in the HI literature that is published. So this will be looking at where the registry has some strong information and where we could potentially look to get that information published in journals, medical and research journals. You've heard Julie talk about the Rare as One program through the Chan Zuckerberg Institute that we are working on. And for us, that is the development of a collaborative research network where we bring patients and researchers with all types of specialties together to develop a prioritized research agenda. We want to continue supporting clinical trials for new drug development. We had gotten a question in earlier before the conference about how do we do that. We do that because these clinical trials, as they're beginning to decide who they're going to test and use with their new drug, have inclusion criteria that we can actually apply in the registry and find a list of those that might be appropriate. At that point, the way that the registry works is that we would send those participants information about a clinical trial that they may qualify for. And when they receive that type of notification, then they can choose to reach out and see if they want to participate. Ty referenced something we are doing, and it will actually take a little too long to describe in the short time that I have, because I do want to leave a couple of minutes for questions, and that is we are working with a company or an organization called CPATH through our relationship with NORD to share de-identified information along with other academic and research sites also looking at HI and kind of putting all of the information from these different sources in one tool to see if there are any trends or any things that may help us better understand HI when we pull information from multiple sources. All right, and the last one. We talked in a couple of different spaces where this is patient-driven research. This is the HIGR, it represents kind of step one into allowing the patients to have a voice into research. The Collaborative Research Network is the next one, to have a voice into setting a prioritized agenda. So what I have for you all is, what kind of questions about HI do you want to see us reporting on through the registry? What do you want to, what other types of questions do you feel like we should even be asking in the registry? It's not a static, it's not a one-time thing. And the last bit is, how do you see HI families involved in HIGR's work? So those are the things that, there's a lot that we've asked of you over the last couple of years, where I want to kind of turn around and say, now is your turn. The registry is strong and rich, and we want to hear from you. And I know we are running short on time, but I want to let you know that these are the questions that we have for you, and look forward to hearing from you in the near future about how you would like, information you would like to see, and how you would like to be involved.
Davelyn, thank you so much, and Ty as well. That was a wonderful presentation, and we have lots of information on the website about joining the HI Global Registry, learning about the HI Global Registry, and we have the video that we tried to show on our website. So it was great. And now we're going to move on, and we are going to show you HI stories. And HI stories are hearing directly from the HI families. We have a family from the United States and a family from the UK who will be sharing their experiences with you. And tomorrow, we're going to have all 5 videos that will be full length on the conference website, so you can enjoy all of them and get a more in-depth view to each of the families that we've shared with you. We're ready.
So the type of HI that Zori has is unknown currently. They did genetic testing, and there was a area of variant unknown significance for the GCK gene that I had and that she also had.
As far as the biggest challenges go, I would say mainly were all the medications involved initially, and having to actually prick her heel 4 times a day to start.
It was 7 when we started.
So many times, but just to have to prick her little baby heel, and then managing the medications and getting the right dosages, just getting that all under control. That was one of the biggest challenges initially. But as far as the triumphs go, definitely we were very excited to be released and come home.
Absolutely. That was probably our biggest
finally
triumph.
Get home when we were expected.
I think just the unknown, like we didn't know anything about hyperinsulinism. So we were like, is our baby gonna die? Like, I think that was probably the way that it definitely negatively impacted our family, because we didn't know what to expect. We didn't know what was gonna happen.
It definitely made us stronger, definitely more resilient. Just having those many challenges, and as my wife stated, just not knowing if our baby was going to die in the beginning. We had no idea how serious of a condition this was. And
it's just like having a best friend. It sounds so cheesy to say, but like having a best friend to kind of hold your hand and lead you through.
Hi, we're all Lee family from Brighton in England. I'm Trudy. I'm Janet. I'm Connie. I'm Ruth. And this is Hattie. You've got HI. HI.
No, it's good. Keep going.
I've got the diffuse form. I am currently on Nanglutide. I am diazoxide responsive.
And you wear a Dexcom.
And I wear a Dexcom.
And you still have hypos.
And I still have hypos.
And you're an adult now.
And I'm 21.
You're 21.
Yeah, 21.
Yes.
What is the best bit of having HI?
Having Connie.
Here, here.
Yeah. Yes, we adopted Connie, and because we're both nurses, we said that we were willing to welcome a child into our family who had a health need or a disability. So if it wasn't for HI, we wouldn't be the family we are. Yeah.
And Connie once told me, after she'd got to sit in the driver's seat of a Brighton bus,
Woo!
that being disabled was wonderful because it gave her lots of opportunities that she wouldn't otherwise have. She said a similar thing when she came back from her tour around America. Connie, what's the tricky bit?
The tricky bit would probably have to be the low blood sugars, because that stops me from doing things that I want to do sometimes.
I remember when you turned 18 and we tried to take you clubbing, and we spent the whole night.
My 18th, no, not my 18th birthday, that was time afterwards.
Was it your 19th?
No, it probably was my 18th, you know.
I remember when it was your 18th birthday and we tried to take you clubbing, and we had to go up and down every chip shop in Brighton because of your hypos.
Yes.
Yes.
It's from all of those sugary drinks.
As well as having HI, I have a brain injury, which does randomly affect my word finding. Yeah, my word finding.
How about you two? What was it like for you two? Connie having HI and having a sibling, and Ruth, what was it like for you having a sibling with HI?
I think having a sibling with HI was really special because it taught me very, very early on about being kind and being very, very grateful. It felt like that about 5 times a day we had reminders about how lucky we were that Connie was still here. And I think, I don't know, I said to another child when I was younger, didn't I, that it always seemed really strange to me that other siblings fought, because I was like, but my sister nearly dies 5 times a day, and you have to, you can't be mean to each other. That doesn't make sense.
And what do you think was different about your childhoods because of HI? Vomit?
Yes. Not a small amount of vomit on the sort of carpet, clothes, dog, sofa, down the back of my pajamas,
In the car,
in the hat, in the bus. Yeah. Connie had a doll with a gastrostomy.
Yeah.
That was different. We always had rusks.
Rusks.
And we always had the what were they called? 40 sip, the drinks that were a meal in a drink and were the reason that until about the age of 14 I was very round.
Whenever we went on holiday, the largest suitcase just having Connie's emergency stuff in.
Whenever we went on holiday, going through customs and having to argue that apple juice counted as a medicine.
Oh my word. We still have to do that.
We do still have to do that.
Yeah. Even though we've got a letter. We have to do that soon.
Official doctor's apple juice permission slip.
What does Congenital Hyperinsulinism International mean to us?
Well, it's well, to me, it's like a way that I can connect with other people with my health condition. So I have contacts from about, say, 5 places, 5 or 6 places, who I talk to from this group, which is a rather good way how to make friends, in my opinion.
For me, it feels like the place where we're the same as everybody else, where we can see people where we don't even have a language in common, but where we can look at each other and go, we're the same. Where there are people from different cultural backgrounds, different religious backgrounds, but who are the same.
Well, for me, yes, we've met some absolutely wonderful, lovely people, and we're all walking in the same shoes. So we, there's that, we know. Not literally, Connie.
I remember when you first found the HI Facebook group, and you called us in. You were like, "Look, they've got a Connie! Right, look, that's a Connie!" And it was so funny because we just truly didn't know anyone with HI.
Yeah.
Anywhere.
It's really affirming, and it's just lovely, seeing people who look like you, who are like you, some younger, some older, just making friends, just making connections, and meeting people like us, fighting the fights we fight, celebrating the celebrations we celebrate. Yeah, so that's us.
Say bye.
Say bye-bye.
Bye.
Thank you so much for joining us for our morning session, and to those of you who helped tell the HI story in the videos. So it is now 12:21 Eastern Standard Time. We will start our breakout groups this afternoon at 12:45. So we're going to try to get back on track on time, so it will be a little shorter break than in the schedule. Just so you're aware, you can enter early, and you will do so by clicking onto the group through the auditorium, the same way you joined your sessions this morning. As Julie said, you'll be able to pick whichever one you would like. The only one I will mention, the medication treatment access group, is for clinicians, and other individuals, industry individuals, and that sort of thing, but the rest are open, and we encourage you to join us a little early. They'll start at 12:45, so feel free to start joining as early as 12:30, 12:35, and we can't wait to see you then. Thank you so much for a great morning.